Thank you very much for the introduction, and thank you for the invitation. It's a real pleasure to be here in my home country after a few decades in the US, and it is a privilege to give this talk, not only as a invitee of the meeting, but also as an EMBO lecturer. It's my honor and great pleasure. So I would like to take the next, let's say, 45-50 minutes to briefly introduce what we call, what is a theme we have been promoting lately. I'm from physics engineering background, but I have to acknowledge that in recent years, we have seen how structure-based models that originate in physics and engineering concepts have been very useful in predicting dynamics. But what we have also seen is that when we combine them with machine learning methods, they become even more useful. So that is the theme I would like to present, show how knowledge of structure-based dynamics combined with machine learning tools may provide us with even more information. What we have been doing in the last couple of decades has been to understand the bridge, like many of us, understand the bridge between sequence structure, oops, apparently, and dynamics. Now, with the understanding that, I'm sorry, okay. With the understanding that sequence encodes structure, as we all know, and then each structure has a unique dynamics. We've shown that in earlier studies. As long as you take a rigorous account of the 3D topology of interresidue contacts, then you can have a very good understanding of the collective motions that the structure may undergo under equilibrium conditions, and that is what we call as the dynamics, actually. And we have been also showing with another student, İvet Bahar, who did his PhD with me, that those motions that are predicted encoded by the structure are also functional. So these are the basic premises that have been underlying the use of the simplified models, elastic network models, that our lab has been using. So we have been always emphasizing that structural dynamics is essential to controlling and modulating our target protein's function. Now, you all know this has been really a huge development. When AlphaFold, or its version 2, were shown to be able to predict structures. And you can see here in this diagram, I think I'm not going to use this. You can see here in this diagram how there's an increase in the prediction accuracy in those CASP competitions. Some of you may be familiar with CASP. These are the annual competitions that are being organized for predicting structures. And you can see that in the year 2018, there has been a jump in the usual prediction accuracy of about 40%. And then 2020, it became even higher than 80-90%. So it's a big development. So just to give you an idea, the PDB, the Protein Data Bank, has been founded in 1976. The first 15 years, there were about 1,000 structures deposited, and then since 1993, approximately the last 3 decades, there have been a systematic increase up to 200,000 structures in the PDB. And what AlphaFold did was to deposit in the last couple of years, 200 million structures, 200,000 in 3 decades versus 200 million in 3 years, let's say. So it's a huge event, and now we need to recognize that this is the deep learning that enabled that. But we also need to recognize that what AlphaFold did was to exploit all the data that have accumulated in the last decades and take advantage of this knowledge. So it is, and not only in this area, in general, in structural biology, there have been really seminal works for predicting protein-protein interaction fingerprints, or a better resolution of cryo-EM structures, or even computer-aided drug discovery. We do in both molecular simulations and data-driven analysis that you can see, we know that there have been lots of contributions from deep learning. Now, since such machine learning methods have benefited from structural data to learn and predict structure, the question we have been raising is that according to this schematic, you can see that structure determines dynamics, and dynamics in a sense defines function, which is the main goal, of course, to understand the origin mechanism of function and devise methods for intervention. So then we said, how about using dynamics data directly? Dynamics data that also now novel models, methods enable to have a better understanding of function using machine learning methods. So that is the question that we have been raising. And dynamics data, when I say, could be generated, as many of you know, through simple molecular dynamics simulations. So molecular dynamics simulations are methods that would be generating long trajectories, and it will take perhaps weeks or months to have some representative trajectories. They are extremely slow, very expensive computationally, not applicable to large systems, and can't be used for high throughput computations. So if unfortunately, molecular dynamics simulations, you can use those trajectories to obtain some kinetics data with the help of machine learning methods, as has been done using Markov state models, for example. However, they still are limited by computing efficiency. On the other hand, if we use analytical methods, and some analytical methods, they do lack precision, we know that. However, they still take account of the entire 3D topology, and they provide for each structure a unique solution, and they can be applied at the proteome scale. So that's what we are proposing. And as analytical methods, we're going to use elastic network models. Elastic network models, as you can see here with this pretty picture, or the way we are using the modes from the normal modes predicted by elastic network models, they enable us to visualize what are the most probable conformational changes or dynamics that a given structure is able to do under physiological conditions, native state conditions, and how it might be altered by some external effects in the environment. So this is the plan of my talk, the next 30-35 minutes. I'm going to talk about elastic network models for those of you who are not familiar, and also introduce a few latest applications. Then show how they can be used in machine learning algorithms for predicting the effect of mutations on function, and even the effect of deletions on foldability. So we start with elastic network models. So some of you, again, are familiar. Instead of the usual ribbon diagram, we transform the structure into this type of network representation, where you can see that it is composed of beads and springs. And what we usually do is the first thing we do, we take advantage of known structures. And you can imagine that advances in AlphaFold 2 have been really very precious to us, because now we have the structure for any protein we would like to. At least we know the confidence levels too. So using the structure, the first thing we do is to map it into a connectivity matrix or contact map. And this is the most important ingredient of the theory. Once you have the contact map, some of you may be familiar, if you invert this type of matrix called, different names, Kirchhoff matrix, Hessian, Laplacian in different disciplines, when you invert it, as shown in the last equation here, you can see that the inverse of this matrix scales, the diagonal elements scale with the mean square fluctuations of individual residues. So the connectivity matrix already gives you information on the conformational flexibility or mobility of different residues. And in the same way, you can use the same equation. The cross of diagonal term will give you information on cross correlations, the way different parts of the molecule move, how they are correlated, which is extremely important, especially for allosteric proteins. Now, what is even more important is that this type of representation lends itself to an eigenvalue decomposition, which you can see here. And you can take advantage of this type of simple mathematical transformation to obtain the effect of different modes of motions that we classify as low frequency or high frequency modes. And you can see the low frequency modes as a function of residue index. They are going to give you the profile, how different amino acids move under equilibrium positions, their fluctuation amplitudes. And at the high frequency range, the profile looks very different. You can see spikes like that, which corresponds to regions of energy localization and correspond usually to highly conserved amino acids. So what the network models do is to transform your static structure into a movie like that, in a simple sense. So, and then let's say for HIV reverse transcriptase here, you can see that is the most dominant mode of motion. And now we can start to interpret these observations. For example, some of you may know this region, the red region that's moving a lot, is called the fingers domain. And it tends to move such that it is going to grab the RNA molecule in between. So this is the way it functions. Now, if we place an inhibitor right at the base of those fingers, which is called the hinge region there, it's going to obstruct the motion. And most of HIV reverse transcriptase drugs have been designed to bind exactly that hinge region. So and then more recently, we have others binding the second hinge region, which we predicted about more than 10 years ago. Now, it is important to understand this type of movements so as to be able to design methods for interfering with those movements. And not only the first mode of motion, the most cooperative one, but also there's a hierarchy, and we can go to different types of motions to understand better what is the mechanism and how we can manipulate that. And of course, over the years, we have been applying this to many systems, from molecular machines to membrane proteins, receptors. This gamma secretase is particularly important for Alzheimer's disease, for cutting the beta amyloid fibrils. Or to cryo-EM structures more recently. So those of you who might be interested in generating this type of movies, understanding better the mechanism and downloading all sorts of quantitative data, you can use the PRODI interface that we have developed, originally introduced in 2009, and recently we had a new version also. So what PRODI does in principle is to, once you give a sequence, you can extract from the PDB all the structures that have that particular sequence within a certain sequence identity threshold. And then you can calculate, you can examine those structures. These are we call that experimental data. And on the other hand, we can also, for a representative structure, you can calculate the modes of motions, the theoretical data. You can compare theory versus experiments, and you can make predictions on the conformational space that is being sampled. So lots of different features. And to date, more than 150,000 users have been using it, and more than 2 million downloads. So we are very pleased, and I would strongly encourage you to test and report bugs. And another interface that we have built is called Dynamics. This one has been developed actually for non-computational scientists, non-computational biologists. It is just an interface where you can, submit jobs and just obtain approximately the same type of outputs without any requirement to know coding. Prodi uses Python language. That one is totally intuitive, interactive. So for those of you, again, interested, I would recommend you to play with it a little bit. Now, again, using this interface, you can generate this type of movies. Now, a few important features that are possible to be evaluated with the help of those elastic network models. One of them is the perturbation response scanning my former collaborator students have developed, which is if you perturb a given position, what happens, as the name indicates. So in very, very simplistic terms, actually, we are talking about elastic springs, right? So simple elasticity equation is F equals Kx, force equals spring constant times deformation. That is exactly what we're using, the spring, but in a matrix notation using the connectivity matrix. And it is possible to pre-multiply by the inverse of the connectivity matrix, a fixed force constant, spring constant gamma. And for any given perturbation, you can calculate the response, the change in the position of individual amino acids. So that is the whole thing. If you want, you can define any perturbation that you like, and you can look at the response. That is one feature that we have been using for determining the amino acids that act like sensors or effectors of signals. So if a given site is very sensitive to perturbations, we call it a sensor. It senses very strongly the signals. And then if a given site is a very good, strong communicator of perturbation, we call it an effector. And as I'm going to show you, these two properties in the network representation, the ability to act as a sensor or as an effector, turns out to be an important feature. And in mutations, for example, we would like to consider whether we are interfering with such properties. Okay, another property we have also considered is essentiality. So the that's more recently introduced, as you can see here. It is possible to also identify what are the most critical sites that dominate the overall dynamics, the overall motions, which, again, to us, dynamics maps into functions. So these will be critical if you would like to maintain the function. You don't want mutations at those sites. So this could be orthosteric sites, active sites, or allosteric sites where you have something binding at a given allosteric site, you have an effect elsewhere. That's very important for signaling proteins. Or, as I mentioned, hinge sites, which are at the interface of domains that undergo coupled movements. Okay. And then using this type of essential site scanning analysis, we were able to identify, generate this type of profiles as a function of residue index, determine what are the most critical sites, generate color-coded diagrams showing the locations, and not surprisingly, in many cases, those essential sites are the drug binding sites, because they are, in fact, altering the dynamics if targeted. And we have seen, for example, in an application to GPCRs, the family of GPCRs, the regions we identified and color-coded by red to be essential sites, they turn out to be generally drug binding sites, and even in this case, it's the G protein binding site. So when nature selects to interact, it selects an interaction site for two proteins, it ought to be consequential, right? So these essential sites that would be inducing a change in conformational dynamics can be identified very efficiently by this method. Okay, so that is the basic. Now, this one shows the extension, something that we have recently reviewed. So, sorry for the very complicated slide. At the center, you have the basic methods like Monte Carlo, energy minimization, etc. And I included the elastic network model-based normal mode analysis as the basic method. And then at the periphery, you have the hybrid methods that use combinations of these basic methods. So, and then you can see from the arrows which one is using which pair. So these are several different studies from the literature, all of them showing elastic network models, normal mode analysis, in one way or another by combining full atomic molecular simulations. So the field has really exploded, and there are lots of such studies. And for those of you who are very interested, you can see the review that we have recently published on such hybrid models that utilize elastic network models or normal mode analysis. Now I'm moving to the next item, effect of mutations on function. So that started with a postdoctoral fellow in my lab who has Luca Ponzoni, who has been very systematically trying to bridge between different disciplines. He's also from physics background. And so what Luca did in his original paper in 2018 was to try to predict the effect of missense variance in the sense that whenever you have a variation in the DNA nucleotide, it reflects, it is a change that's not silent, but gives rise to an amino acid change. And for this type of missense variance, we would like to know whether a given missense variant, a given substitution of an amino acid in a protein, gives rise to a pathogenic reaction. Does it impair function or not? So that's the question. Ideally, suppose we have a scoring function of this form for a given mutation, such that neutral mutations obey distribution and deleterious this one. Then for each new mutation, we could look at the pathogenicity score and make a decision, right? Ideally, that is what we want. That is what actually happens if sequence-only properties are taken based on sequence conservation. That has been the most common approach. As you know, when you do multiple sequence alignments, if a given position is highly conserved, you would assume that it's not going to tolerate mutations. So that is, unfortunately, this is how the distributions look like based on sequence only. Now, what we have done is the following. Now, most of the approaches, again, use sequence-based features, conservation, substitutions at those positions, etc., substitution matrices. And then many others in recent years, they also incorporate structural features, not necessarily the mutant. You can look at the wild type and make assessments. And the most important structural feature would be the accessible surface area. By definition, you would think that something on the surface would be more easily replaceable, right? That's not always true. We're going to come to that. And then we said we're going to include ADNA, dynamical features like the mean square fluctuations, the results from this perturbation response scanning analysis and some information on mechanical stiffness. And we would like to see whether the addition of dynamics features improve the prediction accuracy. Of course, in all these type of studies, the first task, a very, very important task, is to have a data set, right? Now, we're going to have a data set for training, for testing. And in the literature, we found that there are many, many databases, as you can see here. Here, HumeVar predicts SNP, ExoVar, etc. And some of them, and there is some overlap between them. So we found a common set a union of all those sets, which a non-redundant set, which have structural data, because we're going to use structure and dynamics, as you remember. So using this data set, the question was to evaluate the accuracy that can be attained by adding structure and dynamics, understand how much dynamics contribute, and we're not doing anything sophisticated. In this case, it's just a random forest classification trained on 20,000 annotated human missense variants. Now, what we have observed, as you can see here, is that if you take into consideration only sequence or only structure and dynamics, the rock plot, many of you are, of course, familiar with, gives you some improvement over random. But if we consider all three properties, there's a very net increase, as you can see in the sensitivity specificity plots. And we can also go back to the set that we analyzed, and we can see how sequence-only classifier and structure and dynamics-only classifiers give different distributions. And if we include all of them, this is much better now. This has been introduced and used in an interface called Rhapsody for rapid high accuracy prediction of single amino acid variance outcome based on dynamics. We had some difficulty finding a good name, but Rhapsody turned out to reflect what we're doing. What Rhapsody does is to generate this type of saturation mutagenesis plots in silico. As you know, this is becoming more and more now widely used, this type of saturation mutagenesis data. Now we can have a first estimate. What does this matrix display? The abscissa is the amino acid number, and the coordinate is the amino acid type, one of them here shown in yellow being the wild type, and the color code from 0 to 1 will tell you the probability of being pathogenic, 1 being the worst, and in this case, 1 is fully red. And you can see some amino acids are all blue. They can tolerate any type of substitutions. Others are almost all red, and then others depend on the specific substitution, etc. And what we have been plotting here in the bottom is the behavior, single amino acid behavior, irrespective of the substitution. So some positions are really very critical. And we've been comparing this with predictions from other tools, which are shown here, Polyfan2, EVFold, etc., which are shown here. And this was in 2018, actually, or 2020, I think, the comparison with existing methods, where we have 183% accuracy. At that time, we also found out that there was an additional 5,000 variants that we had not originally included from CleanVar, and then we repeated. So this was a good test, and repeating the same calculations using the existing tool, we found that, again, the accuracy is about 0.82. 82% of cases are predicted accurately. So that is what I would like to emphasize here. There may be further improvements using especially what is being done using coevolution of amino acid lately. This is really a breakthrough. However, whichever method you're using, our point is that it is probably going to make an improvement, a contribution, if you incorporate dynamic properties. Now here, you know, random forest allows you to evaluate the weight of each property. Entropy, Shannon entropy, etc. These are not surprisingly dominant effects. But look at the last four, mean square fluctuations, effector, sensor, and stiffness. You can see very easily that they have substantial contributions, okay? And these are the different types of metrics which we show. And I'm going to move to the last part, effect of deletions on foldability. So far what we were doing is, you know, we use the data, learning data, whether a given amino acid substitution has been observed to be pathogenic, you know, disease-causing or not. Now we are focusing on the stability, when we say foldability, whether the folded structure is being maintained. And in this case, we are considering deletions, indels, okay? Insertions, deletions, not amino acid substitutions. So the question we raise is the protein going to retain its original fold in the presence of deletions? And these are not necessarily single-point deletions, which are usually inconsequential, but stretches up to 23 amino acids in this study. And we are now further pushing it. So now you can see this example where on the left you have a loop, which has been deleted here. And despite this deletion, the protein seems to be retaining its fold, as you can see. So the question is, can we predict which particular deletions will be tolerated, accommodated by the fold, and which ones aren't going to be accommodated? The thing is, there is no computational framework for that. There is no existing data set. There was no existing data set. And then we have been generating that. So in this case, we have very few incidences, what we call the positive incidences, the cases where an amino acid, a deletion, has been observed to retain the fold. So this we know for sure because in the PDB there is a structure for the wild type and for the deletion mutant. So there are such cases, about 87 plus 66, which we call positive cases, where the deletion does not make a difference in the stability. And then now we would like to augment the data. So we have been using a series of unlabeled data. So this is this approach is positive unlabeled learning based on, again, random forest classifier. These are the features that we have used. You can see here again the sensitivity. Sensitivity means being a sensor or effector or fluctuations or essentiality that I mentioned. So for the given deletion, let's say 20 amino acids, you may have different values for the essentiality of different amino acids that are deleted. We take the mean values and the maximum, minimum, etc. So that's why we have 6 features. And then what we call profound is from an earlier study where it's mostly sequence properties and structure. Okay, so when we start to analyze, you know, the z-scores corresponding to these different features, you can see immediately, especially in the effectiveness acting as an effector, the difference between positive and unlabeled subsets. And you can, so we can already view that the dynamic space features, they do discriminate between positive and unlabeled multiple amino acid deletions. And then if we look in more details, for example, focusing on effectors and different substructural regions, loop, non-loop, or everything, we can see again the differences. So what does it mean? Let's try to interpret that. Signaling effectiveness is lower in the case of positive in the mutations, which means those loops or other regions that have been deleted are not as involved in transmitting signals as others in the unlabeled set. So we have to, we can see that the difference. So we don't want our amino acids lying in the deletion segment. We don't want them to serve as effector of signals. And then if we, based on this type of simple analysis, we can see that we have a recall rate of 78%. So about 78% of the positive deletions are predicted accurately. And this 19.8% is also interesting. Among the unlabeled case, we have about, let's say, 20%, which turn out to be also positive, which would be tolerated. So here we are listing again the same properties that I have already described. And here's a comparison of what we can obtain with only dynamics properties, just Prodi predictions, different features, and you can already reach this type of 78%. If you introduce additional properties from profound, you can reach up to 84.3% accuracy. Now, if we look at the contributions of the properties, 6 dynamics and 41 others, we can see that the dynamics property at the loop regions, they account for almost 70% of the prediction. And I'm sorry, in the case of non-loop region, it's close to 80%. And then we just conclude that structural dynamics is the major determinant of fault tolerance to adaptability to deletions. Okay? So in conclusion, we can say that intrinsic dynamics is a major property, major determinant of the adaptability to deletions. And these are all features that I have already described. You can reach currently up to 84.3% accuracy, and 20% of random deletions have no destabilizing effect, etc. Now, what is missing? How can we further improve? In all those studies, both the amino acid substitutions and deletions, what we haven't done is what happens when the molecule fluctuates between equilibrium conformations under equilibrium conditions. So, for example, if you take here Groyal interface between the upper and lower rings, the ring structure, you probably know these are 2 heptamers that undergo counter-rotations, as we can see here. Now, a given substitution originally, which might look innocent, might have a strong effect because it would have now a different role in the newly reconfigured conformation. So in the wild type, it might be fine, but we know that the molecule undergoes this type of conformational rearrangement, and now the substitution may encounter an opposite charge amino acid and could get stuck there. This is exactly what has been experimentally observed. So it is important to not only consider the wild type amino acid, but the alternative conformers that are highly probable and what will happen under those circumstances. Something we haven't yet done. Another important feature, when we look at the false positives, what we have observed is that in many cases, some amino acids located on the surface, which are predicted to be neutral, are not. So the thing is, this is all distorted, but the idea is that a given surface amino loop, for example, which you can think could accommodate substitutions, is not going to accommodate if this loop is particularly involved in recognizing another protein. So substrate recognition sites are important. So what we have started lately is a systematic study of protein-protein interactions, which already exist in the literature. There are excellent studies, including those from the developer of PRISM here. And we have been using this type of information to include in our predictor, and try to see whether there is an improvement. So that's the new direction. So we can learn from the, so in some cases you can have the inactive form being locked, or in you need to worry about the involvement of those amino acids in different in different assembly complexations, multimerizations. So in conclusion, I would like to say that, again, structure-based dynamics are very important, and they are even easier now with the availability of structures enabled by AlphaFold. It is not a black box. That's what we also like. We can go back and try to see, what went wrong, which particular properties are really dominating the observed behavior, so that we will have a more rational design of interventions. And then the future directions, as I mentioned, is protein-protein interactions, and also consider ensembles of proteins, families of proteins, to consolidate our results. And we are using increasingly larger structures and networks with known topology. So in the last 2-3 minutes, I just wanted to mention the fact that the elastic network models are applicable to many systems. Recently, we applied this to gene-gene networks, for example, recently published a few months ago. And another application has been, in our case, to chromosomal dynamics, so enabled by high-C data. So you can see high-C data would give you information on contacts between gene loci. And if you enlarge a small portion here, you see the same behavior. There is some kind of hierarchical level of organization, and a type of organization that repeats itself at several levels, a little bit reminding us of the way Fern is designed. You have the same structure being repeated at smaller scales as well as larger scale. These concepts of fractal geometry of nature by Benoit Mandelbrot. So network models are ideally suited, and they can scale up very easily. And that's why we have been also using them for understanding chromatin dynamics, entire chromatin. So that is something I wanted to emphasize. So you can, using the high-C models, using methods that have been developed in structural biology, constraint-based, then you can generate models, 3D models for the entire chromatin for all the 23 chromosomes. But then this type of models, and the models are quite broadly distributed, but still maintain key contacts from high-C. And then using these models, no matter what the size is, it could be easily tens of thousands of nodes in our elastic network model, we can predict what are the collective motions of the entire chromatin. I think this is a really important thing to understand, that DNA is not a 4-letter alphabet, not even a 3D structure, but a 4D entity, including time. And then you can also calculate the fluctuation profiles for different gene loci. So this is just, we have applied to a series of different cells, and then we can show that the fluctuation profile, in this case shown in yellow for the lung fibroblast IMR90 as a function of gene loci. You can see there are peaks. That is for, I think that's for one of the chromosomes, as far as I remember. And you can see the departure from the average behavior of, at that time, 16 cell lines, cell types that we considered. So the difference is in green. So this type of differences in fluctuation profile with respect to an average prototypical behavior of any DNA could be used for rationalizing, for understanding better the gene expression or the cell differentiation. So the question we raised at that time was, does the mobility profile of different gene loci correlate with the regulation, upregulation, downregulation, or even cross-co-regulation of different genes? And here's a simple test that was published about 2 years ago, where we have used the data collected by Abi Mayan, and where you have the information on gene expression profiles. And we would suppose now these are the gene expression profiles for different chromosomes, for different cell types, and we run them through a filter where suppose we know the gene expression, gene fluctuation profile, which gene loci are subject to large movements in the space, which ones are more constrained. And then comparing this profile, mobility profile, with the gene expression profile, we can filter out exactly the correct gene. It means, for example, if this filter is the one corresponding to IMR 90, and we have many cells, we can detect, based on their gene expression patterns, which cell this corresponds to by this type of Jacquard plots. So that's, to us, a very interesting direction. We have done, this analysis was for genes. You can do for the non-coding regions as well. And I would like to conclude with a few final remarks, saying that the generation of elastic network model is very fast, and that's why the outputs could be used in machine learning or deep learning algorithms. It's applicable at the proteome scale and can bridge between macroscopic and macroscopic properties. This is a review we wrote very recently. As the title indicates, it's talking about the mutually beneficial confluence of structure-based modeling of protein dynamics and machine learning methods. And this is a figure from that review, actually, where we are talking about microscopic properties on the left. Microscopic, like the spatial covariance, or the ensemble of conformations, the spectrum of motions, the allosteric signaling mechanisms. These are all microscopic molecular properties. And on the right, you have some observables, like the pathogenicity, the kinetics that are measurables, or binding affinity. So it is possible, as we show in many examples here, to move from that space to that space by combining the two disciplines. And I would like to conclude by thanking many lab members. All this could not be possible without the work of my lab members. And I have been very lucky to have many collaborators. And I apologize for not being able to list others. I have to acknowledge the support from several NIH institutes, as well as the Human Frontier Science Program. And here is one of the most recent photos we took. During COVID, it got more difficult to have group photos. And finally, EMBO would like to remind you that there is such an organization, European Molecular Biology Organization, where the goal of which is to promote excellence in life sciences. And we should nominate many of you for membership. Postdocs, you can always, or new young scientists, you can apply for fellowships. They offer courses, workshops. The EMBO journal or EMBO reports are very prestigious journals. And thank you very much for your attention.